Okay, so remember today we are having lab, and you want to bring your lab notebook to lab because you're going to turn it in today. And so you should have the lactase persistence homework done. So here's a handout. So you want to make sure that you put that into your lab notebook, and then along with the lab we're going to be doing today, you're going to turn it at the very end. I'm going to try to have it graded and turned back to you, if not this week, but beginning of next week. Okay. Are there any questions about the lab notebook? No? Okay, also remember that we have a quiz, but this time it's going to be on Wednesday. So the quiz is going to be, the third quiz is going to be over the information since the last quiz. I'll hand out back the last quiz on Wednesday as well. So you want to make sure you come on Wednesday to take that quiz. We were talking about the structure of eukaryotic cells. And remember that eukaryotic cells are larger than prokaryotic cells, and they are partitioned so that different parts of the cell do different things. And so, for example, we talked about the mitochondria, where we produce energy. And remember, one of the unique characteristics of the mitochondria is that the mitochondria has its own DNA and is capable of reproducing inside of the cell. And do we get our mitochondria from our mother or from our father? Mother. Right? So mitochondrial illnesses sometimes are passed down, or variations or traits are passed down from mother to offspring in a patrilineal line rather than mixing of the genes between males and females. Okay, so are there any questions about, oh, almost out of, are there extra handouts anywhere up there? Do you have an extra one? Okay. So are there any questions so far about the structures that we have gone over in the cell? No. Okay, so we were specifically talking about the endoplasmic reticulum. So this is the endoplasmic reticulum. One of the things is it's near the nucleus, and it has these dots. And if it has the red dots on it, it's studded with proteins. And those proteins are called what? Ribosomes. And so ribosomes specifically are responsible for the production of proteins. We're going to talk about that in a lot more detail when we get to molecular genetics. So that if this cell is going to secrete a substance, specifically secrete a protein, it is going to be produced in the rough ER. Okay. So if we look at another diagram from your book that shows this structure. This shows the endoplasmic reticulum, the rough version of it. Oops. Let's see what I'm doing here. Sorry. Okay, there we go. So the green little structure here is a protein, and the protein gets produced in the rough ER, but then it gets transported to another organelle. And sometimes this organelle is said to look like a stack of pancakes, because that's the way it looks like through an electron microscope. We're going to be looking at cells that you obtained from yourself today underneath a light microscope, and we really can't see these structures using a light microscope because of the resolution is not fine enough to see the detail. So this is the Golgi apparatus right here. And so you'll notice that this protein gets taken up by the Golgi apparatus, and then it gets modified. And so, like, for example, sugar could be added to the protein, and then it gets packaged in a vesicle, and then the vesicle is transported to the outside of the plasma membrane, and then it releases its substance into the outside, or to the outside of the cell. Okay, so this just kind of shows the interaction between the rough ER and the Golgi apparatus. So if I was to write what the Golgi apparatus, I'll get back to that in a second, the Golgi apparatus function is, Golgi is actually the name of a person. And so sometimes this is referred to as the Golgi body in some textbooks, but in general, it's also called the Golgi apparatus. And so Golgi doesn't really mean anything. And this is where we have the modification of proteins So I'll put modification and packaging. Of proteins to be secreted. Okay. It has another function, and that is that this is where carbohydrates are produced. So carbohydrates would include monosaccharides, disaccharides, and polysaccharides. And sometimes those proteins that are produced by the Golgi apparatus are called glycoproteins. So an example of a product would be a glycoprotein. So glyco, think glycogen, think glucose. So this could be like a lot of little glucose molecules that are bound to the protein. So this is the carbohydrate part of this molecule. So in the Golgi apparatus, sometimes they have sugar molecules attached to the protein. Okay, so this is carbohydrate or sugar attached to a protein. And we're going to talk about glycoproteins. They actually are found also in the plasma membrane, floating around in the plasma membrane, and they're really important in cell recognition, so your body's ability to determine self from non-self. So we'll talk more about the glycoproteins in a minute when we start talking about the plasma membrane. Okay. So if we look at the part of the endoplasmic reticulum that does not contain ribosomes, it is said to be smooth. So this is the smooth ER, or this is abbreviation for endoplasmic reticulum. This lacks ribosomes. So proteins are not produced here. Okay. So the smooth ER functions in the production of lipids. So when we were talking about the phospholipids that make out the plasma membranes, the phospholipids would be produced in the smooth ER. If you're talking about steroids, so if you're talking about the cells that produce testosterone or estrogen, they would have a lot of smooth ER because testosterone and estrogen are lipids. And so that's where those hormones would be produced inside of a testis cell or inside of a cell in the ovary. Okay. This has one other function, and it helps to detoxify substances. So detoxifies substances. And so we talked about another structure that detoxifies substances, and do you remember what that was? What was the other structure? Lysosomes break down food. They're not usually not toxins, but peroxisomes. So peroxisomes would help to detoxify waste products, for example, and alcohol is sometimes a waste product. A breakdown formaldehyde is a waste product of alcohol. Okay, so the liver cells would have lots of smooth ER in them. Okay, so your handout has an example of a very specialized cell that produces milk. And so this would be a specialized mammary gland cell. One of the things that's interesting about the mammary gland cells is that they're found in both males and females. So males have nipples, they have mammary glands, but they never produce milk, right? So they never have the hormones that turn on the genes that would produce milk, but they do have mammary gland cells in them, and they would pass the genes for milk production on to their female offspring, right? So, so we have them, males have them, but they're not turned on. So in females, when the hormone prolactin, you don't need to know that, but when prolactin is produced during pregnancy, it causes the milk glands to mature, and the milk glands are just specialized sweat glands that produce a very complex substance, which is referred to as milk, right? So we would say that this is complex organic mixture of stuff, okay? So when we look at proteins, if the mammary gland cell is going to produce proteins, proteins are coded for in the DNA. So I mentioned before that the DNA is stored in the nucleus, but it can leave, that genetic information can leave in the form of a messenger RNA. Right? The messenger RNA then goes to the ribosome in the cytoplasm. And the information that is copied or is found in the DNA is then transferred to the production of a protein. So that's what we say when the DNA codes for proteins, okay? That's what we mean is that the information in the DNA gets sent out to the cytoplasm, and then that information is read, and then we produce a specific protein based upon the genetic code, okay? So a specific protein is produced. So if we look at the protein in milk, we can sometimes extract the protein by causing it to denature. So has anybody made cheese here before? Okay. So what you do when you make cheese is you acidify the milk. And so this is actually the same thing when you make curds, right? Or when you make yogurt, it causes the milk to become acidic. So you can add like vinegar or lemon juice to the milk. And what happens then is the proteins come out of solution and they become a solid. And so the protein in milk is called, the vast majority of protein in milk is called casein. And so this is the curds. Okay. So when we curdle milk, that is when milk goes bad, it becomes acidic because of bacteria reproducing in it, and the milk starts to get chunky, right? Okay. So that's the milk protein. That protein can be used by the newborn, right, to produce, it has all the essential amino acids in it that it can be used to produce protein in the growing newborn, okay? Now we also have other proteins that are important. So for example, in breast milk specifically, we have antibodies. So antibodies are also proteins. So this is under proteins. Casein and antibodies. Antibodies have an immune function. Okay. So this is sometimes referred to as passive immunity. So the baby gets antibodies via the breast milk, and those are antibodies to bacteria, for example, that the mother has been exposed to during her life. And so she's producing antibodies. And so this is going to have some immune protection. So it protects the newborn from infections that the mother has been recently actually exposed to. Right? So antibodies are a part of breast milk, okay? When you look at the first part of breast milk, the first breast milk, does anybody know what the first breast milk that comes out is called? Colostrum, okay? So colostrum is what is referred to as the first milk. And it is kind of interesting looking because it doesn't look like milk at all. So when you first start producing milk, and you're a female, you produce it, and it looks yellow, and it looks really thick and viscous. And the reason why is that the colostrum has lots of antibodies in it. Okay? And so as your baby develops, the breast milk composition will change, and so it'll actually get more fat. But the first milk is high in antibodies. Okay? So it doesn't look white, and it's not really, it looks more yellow, and it's kind of yellowish clear color, okay? Okay, so those are the antibodies. We also have enzymes in the milk, which help to digest the milk. Okay, so that would kind of go there, enzymes. And then we have things like hormones.
So colostrum is an enzyme?
No, colostrum is a protein. It's actually the first milk, which contains a lot of protein, including a lot of antibodies. But in the milk itself, there are enzymes and there are hormones. Okay. So if you look at the handout that I gave you, and I don't have a copy of it in front of me because I handed them all out, but if you turn it over, and there's a comparison between what breast milk has in it and what formula has in it. And so you see that breast milk has lots of stuff in it that formula doesn't have in it, including antibodies, including hormones, and including enzymes. Okay, so those are the proteins.
So enzymes, so you're saying enzymes are a type of protein?
Yes, they're a type of protein. Yeah. So casein, so in the milk, there's casein, antibodies, enzymes, and hormones, and those are all examples of protein. Thank you for that clarification. Sorry about that. Okay. So we also have carbohydrates. So you should know, what's the major carbohydrate in milk? Lactose, milk sugar, right? Energy. Babies normally have lactase, which is an enzyme that helps to digest lactose. And I mentioned previously that as we develop, we lose the ability to digest lactose. And so most of the people on the planet are lactose intolerant as adults. Sometimes babies can be lactose intolerant, just genetically, and so then they have to be fed special formula because breast milk would make them sick.
Okay. I have a question. So if the lactase, so if you are producing, you keep having that lactase enzyme to be able to break down the lactose, if you continue to drink milk, could that be part of why you see a difference?
Yes, and so what happens, if you can imagine the first people that moved up, the hypothesis is they moved up to Europe, right? And you're starving. It's the middle of winter. There's nothing to eat. So you start eating milk, right? And you get diarrhea from it, and it makes you really sick, and you die, right? But if you drink milk and you're like, oh, I'm just a little bit sick, then you have tolerance to that, and you can use the energy and you don't die. Then you pass those genes on to the next generation. And so over many, many generations, you develop this population that can use lactose as adults. But if you weren't able to, those first people, if they moved up there, if it caused them to have diarrhea and that was their only source of food, they probably just died. They starved to death.
I get that, but I'm just wondering, did that enzyme stop getting produced because people stopped drinking it?
Yeah, so if you look at, we're the only ones that, so if you look at mammals, other mammals do not have herds of dairy cows, right? So other mammals can only drink milk when they're babies because there's no source of milk outside of being baby. So once they, so once you get weaned and you're a mammal, like a cat, there's, you would never drink milk. That's why it's so bad to give them milk, because what we're doing is we're making them sick. But we would, they would never have a source of milk. If they did have a source of milk, then they might be able to evolve lactose.
Yeah, that's what I was wondering.
Yes.
If your body would keep producing it, if you never cut it off, if you kept.
Yes, and but see, we're the only people that
your body keeps producing it. There's a need for it.
Yes.
This idea.
Yeah. I'm just imagining other species having dairy animals. The ants, have the aphids. So ants actually farm aphids, and the aphids suck on the plant juices, and the ants come by and they rub the abdomen of the aphids, and the aphids will actually exude sugar water. So actually, ants are technically milk aphids, but it's not milk, it's just sugar water that they're exuding. Yeah. Okay. So carbohydrates. How about lipids? What type of lipid would you think is in milk? Saturated, right? So this is triglycerides, and specifically saturated fats, right? So saturated, meaning that they don't have any or very few double bonds between the carbon atoms. So they tend to be solid. So breast milk, when you put it in the refrigerator, the milk comes to the fats come to the top, right? That's the cream. Sometimes if you buy full fat yogurt or full fat, yogurt, then there's a layer of cream on the top, and you can eat that, but it's better to mix it in, right? And so it will float to the top because it is a solid under like conditions in the refrigerator. Okay. So one example of a lipid that's important is called a long-chain fatty acid. So we call them long-chained. This is on your handout. And one example of this is DHA. Where have you seen DHA before? Think grocery store when you're shopping. Do you ever see that word? Yeah. So some of the milks, they have DHA added to them because this is believed to be a really important fatty acid for brain development. So this is important in brain development.
Is that just vitamin D?
No, DHA is a lipid. So unfortunately, it's the expensive milk, right? So you go to the organic section and you have organic milk. Well, that's already really expensive, right? But then you have organic milk with DHA added, and that's like $5 a carton, right? Okay. So the thing about the DHA is you'll notice that long-chain fatty acids are not found in the formula, right? So there's not this in the formula. And so they think that could be a really important nutrient for babies. It's found in breast milk. We can add it to milk, right? They don't tend to add it to formula. I don't know if I should look. We've actually have formulas with DHA added to it. Right? Okay, so then we can talk about another lipid, which is cholesterol. So cholesterol is a steroid, and it's an important precursor to producing other steroids. So it's important for producing vitamin D, and it's important for producing estrogen and testosterone. Now, there is something else that is in milk that we oftentimes don't talk about, at least in this country. It's kind of a big issue in other countries. And this is what is referred to as persistent organic pollutants. And these are sometimes abbreviated as POPs, persistent organic pollutants.
Are these also lipids?
Yes, well, actually, these are lipid soluble. So they do tend, they're actually not lipids, but they're lipid soluble. So they're lipid soluble. And so that means they get stored in fat. These are synthetic chemicals. They're not natural chemicals, so our body doesn't really understand how to break them down. So they get stored in our fat, and they just tend to accumulate over our lifetime. And so some examples of these, they usually use acronyms, so like DDT, right? This is an insecticide. It is a persistent organic pollutant, right? And in the 1960s, Rachel Carson was the scientist that pointed out that eagle populations and other large bird populations were dying off because they had so much DDT in their body that they were getting sick, but they also were producing eggs that had such weak shells that they were unable to hatch. So we stopped using DDT the way that we were using it in the 50s, which was just kind of dousing everybody with DDT to kill off mosquitoes, right? So you see pictures of people running along, kids running along the beach in a spray of DDT washing over them, right? We now realize that that is not necessarily a good thing, okay?
The persistent organic pollutant is a form of DDT.
No, DDT is an example of a persistent organic pollutant. Another example is dioxin. And you might have heard of dioxin because this is a chemical that is produced when we bleach stuff. So like when we bleach diapers, or we bleach Tampax, or we bleach paper. So like the white paper, the toilet paper that you get, that has been bleached. And so dioxin is another persistent organic pollutant. And this is why some people are concerned about using bleached paper products near their reproductive system. So for example, Tampax that have been bleached have dioxin in them. So it's not necessarily a good thing to put that up in your body, right? It could be bad. So you can get unbleached or dioxin-free hygiene products. So you can get
bleached flour or.
No, this is bleached with chlorine. So I'll put bleached paper products. Bleached paper products. So diapers that are brown and haven't been bleached are probably better than the bleached diapers. Right? Dioxin is this one. Now there's one that you probably have heard of recently, which is called BPA. What's BPA found in? Water bottles. So this is plastic. And again, it's kind of interesting. This was a female that, I can't remember her name, but she was a scientist who was studying cancer cells. And she noticed that the cancer cells were dividing even when there was no hormone present. And the cancer cells that she was culturing them in plastic tubes. And so she theorized that something from the plastic was leaching into the water and causing these reproductive cells, I think they were cervical cells, to reproduce uncontrollably. And so she was, her story was is that the plastic industry just came after her, just trying to sue her and discount her. And now when you look at plastics, you know not to put them in microwaves because that can be a really bad thing. And you know, because the stuff can leach out, but you know you want to drink out of BPA-free water bottles, right? And they also eliminated BPA from babies' toys because they were thinking that some of that BPA was getting inside the baby's body, okay?
So they do put the DHA in one, in the infant milk formula.
They do?
Yeah, I just looked.
Oh, awesome,
Yes.
awesome. Yeah, so that's been added,
They have two different ones that they put it in.
and it's probably expensive.
Oh, yeah. But they do make it with it.
Right. There's a big, long history of formula, and it one hypothesis is that we promoted the use of formula because it was advertised and marketed, you know, as being freedom for a female, but it's just super expensive, and it's yeah. So think about the difference between breastfeeding and formula. It just really puts it's expensive option. Okay, so these are examples, and what happens is that they accumulate in my tissue or any female's tissue, and we have a lot of fat in our breast tissue. And so we can get these stored over our entire lifespan, and then when we start to breastfeed, they come out. So it comes out in the milk. So breast milk has these types of pollutants in it. And so in Europe, they have what is referred to as breast milk monitoring. And I think, since I mentioned that in Europe, I think that you can now, I think there's probably companies where you could send off your breast milk to get it tested to see if there's a high level of persistent organic pollutants in your breast milk. This is also in cow milk. So I just want to point out that, like, if you're drinking whole fat milk, sometimes these pollutants can also be found in cow milk, which is an argument about why you might want to drink organic milk if it's fat If it's fat-free, it doesn't matter, but if it's got fat in it, organic milk is less likely to have probably these substances. I know it's less likely to have it. It might still have a little bit of it, but it's less likely to have some of these substances in it. Okay, so when we look at an example of how we can clean up breast milk, okay, oops. This is in Sweden. Okay, so it shows some of the persistent organic pollutants, including PCBs. I didn't mention PCBs, but PCBs are like in rivers because they were produced by factories on the edges of rivers. And so they're in like the sediment of the Columbia River, and they get into the food chain so that they're found in like salmon. Right, so if you eat a lot of salmon, you might get PCBs in you. But notice the PBDE. Does anybody know what PBDE is? Probably not, but this one's kind of a crazy one. This is a flame retardant. So PBDE, flame retardant. So
other pollutants, these PCBs.
Well, DDT and PCB, yeah, these are other ones. So PBDE is a flame retardant, and it is put into carpet. It's put into your foam furniture, right? And it was put into kids' pajamas, but they subsequently stopped doing that. And so you can see that over many years, PBDEs increased in breast milk, and then they started cleaning up the environment, and it has decreased over time. And the same thing with DDT. If you decrease the use of DDT in the environment, then you decrease the amount of DDT that is found in the breast milk. So we can have environmental regulation against these kinds of things, and the trend will be down. So PBDEs, they say, if you go to a website, they say, how do you reduce, and you can find this, how can you reduce your risk of being exposed to these chemicals? And one big one is foam furniture. And specifically, I actually had to throw out one of my favorite chairs because the cats had scratched at it, and they had the foam was starting to come out of it, and it was crumbly, right? And I knew it was from a time when they highly dosed the foam in this. And so that is one source, old foam furniture is a source of PBDEs. So they had to go, but unfortunately probably went to landfill. So it's probably in the landfill now, okay? And then maybe into the water. So it's really hard to get rid of them once they get into the environment because they don't break down. We don't have enzymes to break these down because they're synthetic. Okay, so if you think about the breast milk, then you just have to be aware that we can have some of these toxins in the breast milk. One interesting thing is that by getting rid of the toxins, it appears that females actually decrease their chance of getting breast cancer. So if you have breastfed and you're a female, you have a decreased risk of breast cancer because you have essentially gotten rid of all your toxins in your breast tissue. They say you downloaded it to your offspring, right? The firstborn gets most of the toxins. And so the firstborn baby gets most of it, and then subsequently the milk is cleaner. So if you're a firstborn breastfeeder, you probably got the bad share of the toxins, right? So the question is kind of interesting, should we be simulating milk? We could do this. We could cause a female to lactate outside of pregnancy, produce milk, get rid of all the toxins out of the milk before she actually has babies and actually has to breastfeed. Probably the easier way is just to clean up the environment and reduce your exposure to them. Okay, so if you look on your handout, I don't have a copy of that in front of me, but if you look on your handout, it says that this is not something that's found in formula. So they get rid of these, they don't have these things in formula, but there is inorganic pollutants. And so in formula, you have simply nitrogen, right? So nitrogen in water is an inorganic pollutant. Okay, So if you have a well and you want to get it tested occasionally to make sure that nitrogen hasn't leached into your water supply. If nitrogen is present, it's a really bad thing to mix it with formula because nitrogen causes developmental delays, and it can do damage to neurons, right? So you want to make sure that the water that you use is clean. That's another problem if you're talking about third world countries, because sometimes formulas are pushed in, say, Africa or South America, but they don't have clean water, right? They could have bacteria in their water, and then the babies could get sick from that, whereas they wouldn't have gotten sick if they were just using breast milk. So besides it being expensive, it can also, any contaminants in the water, if you don't have clean water, is a problem. So even after all of the bad things that I just mentioned about these persistent organic pollutants, it's believed that breastfeeding is best, right? At least up until up to, what is it, 9 months, maybe a year now, they recommend that females breastfeed. They think it's really important for proper health and development of the offspring. Actually also helps them keep they tend to be healthier, and their immune system tends to be stronger because of breastfeeding. Okay, so if we look at the image of, on the back sheet of your paper, okay. So let's just look and follow some of these substances that are produced by a specialized mammary gland cell. So the DNA gets copied into messenger RNA. So here you can see the messenger RNA is leaving the nucleus. The messenger RNA goes to a ribosome, right, on the roughy R. Then a protein is produced. And notice that the protein is then transferred to the Golgi apparatus where it's packaged, and it moves out and it is secreted, right? So that would be, this would be protein. So the other things include lipids. So let's look at the lipids. So here, the smoothie R, which would be like right here, okay, it doesn't have any studs on it. The smoothie R produces the lipids, and the lipids are just in the cytoplasm, and they can just easily move to the outside. So this would be like a fat, droplet of fat, of triglycerides moving into the milk, okay? Sugar is not shown here, but actually I do show it here. So sugar being produced, this would be proteins and sugars, and so lactose would be secreted to the outside of the cell. So here you can see very much that the eukaryotic cell, like a specialized mammary gland cell, is essentially just a factory that is producing and secreting substances into a duct that connects to the nipple, right, which then comes out of the mother and hopefully into the offspring. Okay. So this is just the specialized version of our body cell. Any questions about this idea? Okay. So we are going to move on, and we're going to talk about the plasma. Oh, actually first, I want to talk about the cytoskeleton. Okay. So I mentioned the cytoskeleton. The cytoskeleton is composed of proteins. And does anybody remember what structure did I talk about that aids in cell division, separating the chromosomes during cell division? The what? Microtubules, but it was called the centriole, right? So the centrioles, so example of this would be the centriole. Okay. Centrioles, so that's important in cell division. And then another example would be microtubules. And these are kind of like railroad tracks. So you could say that they're kind of like train tracks. And they help to deliver and to move substances inside the cell. So substances are moved around inside the cell. I'm going to show a picture. You're going to be amazed. Substances move around like train tracks. Like train tracks.
What is the
Oh, so maybe microtubules like, like train tracks. There we go. Substances move around. Okay. Okay, so we actually have what are referred to as motor molecules. So I'll put by what are called motor molecules. And so you probably heard the word nanotechnology, right? Tiny, tiny molecules that are actually able to move around on their own. And so these motor molecules have been built synthetically, but we actually have nanotechnology inside of our cells. And that is how we get stuff moving around inside the cell. So I'm going to show you just this little animation of what one of these motor molecules looks like. Moving stuff around inside the cell. Okay, so if you look at this diagram, the bottom is the microtubule. That is the train track. It is made of proteins, right? This is my motor molecule right here, right? And then it is connected to a vesicle. So I just talked about how a vesicle can move from the endoplasmic reticulum and the Golgi apparatus to the outside of the cell. And this is how that works. Okay. This is a short video, so you got to catch it. 21 seconds of it. Okay. No, there's no sound, right? That is a motor molecule, okay? That is part of our cytoskeleton. Okay. That is what is happening inside of our cells. Okay. Little train tracks. Okay. Yeah, motor molecules. So we actually have built, they've actually built little motor molecules, and somebody just won the Nobel Prize for it. This one right here. So these just represent atoms. And so we're talking about these little molecules that are just composed of individual atoms, right? So this is a synthetic one that they built that is like a motor car. So it moves, right? So imagine putting something like that together molecularly and watching it move, right? So that's nanotechnology, okay? But we have those inside of our bodies already. Okay, so that's a function of the cytoskeleton. If I could figure out how to get out of here, let's see. Oops. No.
Let's suppose that you're writing a route.
Okay. So the cytoskeleton is a diffuse network of proteins. So you can see here they actually stained the cytoskeleton of a cell. And so in the cytoplasm, we have all of these networks of proteins. And then this just shows the microtubule with the motor proteins attached to it. Okay. And move, they could attach to a vesicle, for example, and move along. Okay, so now we're at the plasma membrane. So we've already talked about the plasma membrane as being composed of phospholipids. So we have the phospholipid bilayer. Okay. Bilayer means that there's two layers. Remember, the tails are repelled, so they're on the inside, and the heads are along the outside. But we also have proteins embedded in the plasma membrane. So we say that these are proteins that are embedded. Now, at one point in time, we didn't realize that these proteins could float around inside the plasma membrane. But now the new model of the plasma membrane is that the membrane is very fluid. And so it's called the fluid mosaic model for the plasma membrane. So it's a way to understand how it works. So fluid means that the proteins can move around, Mosaic means that there is a lot of different, that's IC. Mosaic means that there's a lot of different proteins in it, and they're just kind of like bobbing around inside this membrane. Okay. And they serve different functions. So if we look at some of the functions of these proteins, okay, one is that they can be channels. So a channel means that they can allow substances to move through the plasma membrane, okay? So it allows substances to move into or out of the cell. The second thing that these proteins can do is that they can attach to the cytoskeleton. And they can help give the cell its shape. And we'll talk about a disease called muscular dystrophy, which is caused by a defective protein where the protein does not attach to the cytoskeleton, and so the muscles cannot contract, right? So muscular dystrophy has its basis in cell biology, and specifically a defect in the cytoskeleton attaching to the plasma membrane. Okay. The third thing that this can do is it can give us information about what the cell is. So we can use, we can say that it is, these are the functions of the protein. So this is recognition of self versus non-self. So the proteins in the plasma membrane cause tissue rejection. So if you just try to put any old organ inside of a person's body, that organ is going to consist of cells that have different proteins on them, on the surface. And the body is going to reject them because it's going to detect them as being non-self. And we'll talk about blood typing and how important that is also. So when you put the wrong type of blood in a person, it's because of the proteins on the surface of the red blood cells do not match from the donor to the recipient. The fourth thing is, oh, and I want to point out, this is the glycoproteins. I'll put this as glycoproteins. So sometimes this is called the sugar coating of the cell. Sugar coating of cell. So glycoproteins are specifically self-recognition. We could also have proteins behaving as receptors. For example, hormones can bind to receptors on the outside of the cells. That says receptors on outside of cells. Okay. So insulin binds to liver cells and it causes them to take glucose up out of the blood and store it as glycogen. So receptors are really important. And then the fifth one is that they can be enzymes. And this is actually the example of lactase, right? So lactase is actually an enzyme that is found in the plasma membrane of cells lining the small intestine. So they're found in plasma membrane of cells lining small intestine. So those are some examples. So on the quiz, if I ask you, what are 2 or 3 examples of the functions of proteins in the plasma membrane? This is where you would go to. You go like, oh, well, proteins can function as channels. They can attach to the cytoskeleton. They can be receptors. They can be enzymes, okay? So these are plasma membrane proteins. I'm gonna write that right there because people get confused here. These are plasma membrane proteins. Proteins found in the plasma membrane have these variety of functions floating around in our plasma membrane. Okay. So this is my diagram. So this should be a diagram that you study, right? So if I pointed, and you've probably already seen this in terms of the quizzes online, but if I pointed to that, you would say that is a phospholipid. If I pointed to this, you would say that is a protein. Now, what is this? Oh, this is obviously a steroid, right? This is cholesterol. So cholesterol in the plasma membrane has a separate function than the proteins or the phospholipids. So cholesterol specifically is a part of the membrane, and it maintains the fluidity. And it's kind of interesting because they've done studies on mammals that live in cold climates and hibernate. So if you think about bears, they shut down their metabolism and hibernate, and their plasma membranes are going to get colder than your average because their body temperature is going to drop, right? And they've actually found that animals that live in cold climates have more cholesterol in their plasma membranes, and that helps to maintain a fluid plasma membrane even when it's really cold outside. Okay? So cholesterol is important. We want those proteins to be able to move around. It's important that it's not like solid, or outside of our cell is not solid. It's fluid. Okay. So you notice that some of them have, this would be a glycoprotein because it has a carbohydrate chain attached, right? So this would be like the sugar lining, right, of the cell. And so those are important, cell recognition. The ones that have holes through them, those would be channels, okay? If you don't need to know this, but if they're through the entire one, that is an integral protein. You don't need to know for my quiz. It might be on the quizzes online. This is called a peripheral protein because it doesn't go all the way through. So the peripheral proteins might just attach to this cytoskeleton, for example. But in order for it to be a channel protein, it has to go all the way through the membrane. Okay, so let's look at permeability. So movement across the membrane. So when we look at cell membranes, we say that they are permeable to what can move across just the cell membrane. So one thing that they tend to be permeable to is water. So water can move without the aid of a protein. So we're just talking about the membrane, not the proteins. So we're just talking about the bilayer, the phospholipid bilayer. So water can move through, right? As well can oxygen and CO2. So carbon dioxide and oxygen gas can just diffuse right across the surface of the cell. And that's really important because that's how we get oxygen and how we get rid of CO2 in our respiratory tract. And the other thing that is permeable to this membrane would be lipids. So lipids can be absorbed directly across the membrane. Right? So even our outer skin, which is dead cells, can absorb lipids. And so think about some steroids that you can apply to your body. So for example, you could use progesterone cream. That is a really common thing that people take when they're heading towards menopause, right? Progesterone cream, testosterone cream.
And these are examples of those.
Yep. So these are steroids, they're lipids, and they can be absorbed right across the surface of your skin. So testosterone with an E. You have to be careful of lipid-soluble things that can come into your body, right? So you want to be really careful about toxins that can also be absorbed across the surface of your skin. So sometimes you see, do not handle without gloves because it's something that could move across your skin and into your circulatory system. Okay. Then we say that it is impermeable to, water-soluble substances. So that would be everything besides lipids, right? So proteins, carbohydrates. You cannot put sugar on the surface of your skin and absorb it, right? It's not something that can move across. It can't get across because it can't get through those tails that are hydrophobic. Proteins also can absorb across the skin. So I'm not sure about proteins. They're always talking about these proteins that they add to skin creams like collagen and keratin. You can't absorb them, so I'm not sure how they really help with having them. You can inject them, and if you inject collagen under your skin, you're going to get rid of your wrinkles because it's going to help the dermis. But I'm not sure how you get them into your body if you're just putting them on in a cream. Okay, so water-soluble substances, and then things that are charged. So ions. Okay, so like things that have a charge. So we'll say charged particles cannot get through the plasma membrane. So when we look at these substances, these must use channels, right? And the channels are composed of proteins. So if I want to move sodium into the inside of my cell, it cannot go through the plasma membrane. Sodium is a charged ion, and so it has to go through a channel. Okay. So if we look at how things tend to move, substances will move with their concentration gradient. So even if we're using channels, substances will tend, I'll put tend, to move with their concentration gradients. So I'll just abbreviate that, gradient. Okay, so what that means is that they go from a higher concentration to a lower concentration. So this is like a force of gravity, right? Similar to it's a physical law, a physical force. So substances will always go from a higher concentration to a lower concentration, right? So you can think about putting a sugar cube in water, and initially the sugar cube's all going to be in one place, but if you let that sit overnight, what's going to happen is that the sugar is going to move out until the sugar and the water are equally distributed. So the sugar will move out from center, right, and become dissolved in the water, right? So things come just automatically, without energy, without living systems in our physical world. This is what happens. Right? So if anything other than this happens, that means that we are using energy to make it go the reverse direction, right? So if you can imagine how difficult it would be to take all those sugar molecules and force them back together again into a sugar cube, then you're getting the idea of how much energy it would take to maintain that amount of order, okay, in that particular system. Okay, so the first type of movement is simple diffusion. So simple diffusion is takes no energy. It requires no protein. So it requires So if we're looking at this, what's going to happen is that if we have this membrane and say that this is a substance that's soluble, so what's going to happen is it's going to move across until they are equally distributed, and this is called an equilibrium, right? So they just automatically will diffuse across, right, until they're equally distributed, okay? Now, sometimes we have a type of diffusion which is facilitated. So we can say if it's facilitated diffusion, it still is diffusion, so it takes no energy. But in this particular instance, it requires a protein channel. So it's facilitated by a protein channel.
Simple diffusion ones, they don't go through the protein channel, so
they just go across the membrane, yeah. That's simple diffusion. So facilitated, so simple diffusion would be like oxygen. Oxygen just diffuses into our cells. Okay. Facilitated diffusion would be like if you are transporting glucose across the surface of the cell. Glucose is water soluble, so it would require a protein. It would require being transported. So if you want to absorb glucose across your small intestine, you have to have a protein channel that will carry glucose for you, okay? So if we look at the facilitated diffusion, facilitated diffusion. Okay, so this would be like glucose, okay? So glucose is being facilitated. Its movement across the plasma membrane is facilitated.
So how do you do with the tRNA?
No. tRNA?
Does it have anything to do
No.
with that, with the transporting of it?
Nope. We'll talk about tRNA, but that's totally something different. Okay. Okay. So sometimes we not only just have channels, but we have, I'm not gonna talk too much about this. We have carrier proteins, and carrier proteins will change shape. So they'll pick up something, they'll change shape, and then they let it go. So you don't need to know the difference between channels and carrier proteins. It's not that significant. I'm looking for my one slide I need. Sorry, osmosis, this is what I need. Okay, so osmosis, this is what we're gonna be looking at today. And this is specifically the diffusion of water.
So this is a 30-second movement.
Yes. So we have, it's actually diffusion, so it's a type of diffusion. So we'll put type of diffusion.
Facilitated?
Nope, type of simple. Type of simple diffusion. So this is the diffusion of water. Okay. So in this system, the water can move across the membrane, but the solute cannot. So this is the solute. So this would be like salt, or it would be like sugar, right? It cannot move. It's stuck in on this one side. And so when we look at this, when we look at a solution is composed of a solute, like sugar, plus the solvent, which would be water in this case. So this is water. An example of this would be sugar. Now, notice that what happens is that we actually get a pressure buildup, because water is going to move, and we don't see the water molecules here, but water would be at a higher concentration over here than here. Does everybody see that? Higher concentration of water over here on this side compared to this side, right? Water moves from its higher concentration to a lower concentration. So notice how it will move over, and it is so much pressure that it actually moves and presses against gravity, right? So you would have like this side, if this was a tube, you would have water moving into the tube, and it's actually pressing up against gravity. So that's what's sometimes referred to as osmotic pressure. And this is really important when we start talking about the way that your circulatory system works in the capillaries, and also the way that your kidneys work in producing urine. We talk a lot about osmotic flow and osmotic pressure. Okay. So the confusing thing about these terms is when we talk about the concentration, the relative concentrations of these two different solutions. So we have terms that go with that. So the relative concentration of solutes in two solutions, right? So this could be on one side of the membrane versus other, okay? So isotonic, iso means the same. Think isosceles, right? Think when you put saline into your eyes, when you use a saline solution, this is isotonic. So isotonic means that it is the same solute concentration. So I'll put like a saline solution. So you would not want to put pure water into your veins, right? You would put saline solution into your veins, and I'll explain why in a second. Okay, this is saline, I'll write it better. Saline solution is what you buy. If you have contact lenses, saline solution, okay? Versus, oops, hypertonic. Okay, hyper means above, right? Tonic means the tension, right? So this is higher solute concentration. Right? So we would say, like, if I was to look at this, I would say that this is hypertonic compared to this side. So this side is hypertonic compared to this side, okay? Conversely, we could talk about hypotonic, and hypotonic means that it has a lower solute concentration. Okay, so just like I said before, this side, right, which has fewer, this side, which has fewer sugar molecules would be hypotonic compared to this side, which is hypertonic, okay? So let's look at an animal cell if we put it in a beaker of water. Okay, so this is my beaker of water, and this is my animal cell. This right here is, let's say that we use distilled water. So distilled water is pure water. Distilled water has no solutes in it. Tap water does, but distilled water doesn't. Okay, so this is my animal cell. So notice that these terms that we use are relative. So think about all the solutes inside of your animal cell compared to the outside. So what would the inside be compared to the outside? Hypertonic. The inside is hypertonic compared to the outside, okay? This is where it gets confusing. The outside is what? Hypotonic compared to the inside. So it's all relative. You have to compare one thing to the other, right? So which way do you think water will flow? Is it going to flow out of our cell, or is it going to flow into the cell? Into the cell. So osmosis is going to cause water to flood into the cell, and the cell is going to rupture, right? So if you put pure water into your vein in an intravenous drip, your blood cells would just explode and you would die, right? If you put saline, then water moves back and forth, but there's no net or loss gain, so that nothing happens to the cell. If you put salt water and into your body, it would actually cause the opposite to happen. Water would be drawn out, and our red blood cells get all crinkly looking. They kind of shrink up because they lose water. They become dehydrated, okay? So if we look at what this happens in animals versus plant cells, right? In a plant cell, right, water will move when it is out to the outside. When it is in salt water, so it'll tend to shrink up. Water will move in when it's placed into water. And notice that the cell wall kind of stays the same, right? But that cell wall, there's pressure exerted on the cell wall, which gives it its tonicity. It's being tonic. So when plants dehydrate, they get wilty, right? And that's because there's not enough water in their cells to support their cell walls, and so they just wilt, right? And then all of a sudden you give them water and they go, ooh, perk right back up, right? And so that's just the movement of water into and out of plant cells. But if we look at it in terms of red blood cells, if we put a red blood cell in salt water, so the salt water would be hypertonic, water would move out and our cells would look like that. In an isotonic solution, water just moves back and forth. And in a hypotonic solution, water would move in, causing the cells to rupture. Okay. So that is the significance of osmosis, and osmosis is really important in physiological processes in the body. Okay, so I'm going to stop there for today, and we are going to be looking at osmosis in lab today. So you'll be able to use some of this terminology that we just talked about. So remember, bring your lab notebook to lab. Hopefully everybody has it with them.